Hi everyone, this is Dr. Mamta Chava, and in this video module I'm going to talk about bacterial genetics, the important aspects. Alright, so bacteria give the most thoughtful gifts to each other. Okay, so guys, the only constant thing about universe is change. Alright, and it is very important to change ourselves with changing environment so that we are able to survive. Okay, so it's all about survival of the fittest. So as far as you know, bacteria are concerned, which are so tiny organisms, even they have got this habit of exchanging genes amongst each other. For instance, if one organism has got resistance gene against a particular drug, that bacterium is going to help fellow bacteria around so that they also develop resistance. So just like we help each other, like humans help each other, we help our everybody. Similarly, it is a very wonderful thing at molecular level also to see how bacteria are helping each other. It's a different matter altogether that this kind of help which they do of each other, I mean the bacteria, the way they help each other, that is not going to be good for humans. Okay, because if there is rapid dissemination of a gene which encodes for which has a resistance gene against a particular drug, if there is rapid dissemination of that gene in a community of bacteria, right, and that community of bacteria flourishes and goes on to infect a lot of humans, right, then obviously it's going to be a problem for us. All right, so how is this exchange of genetic material happens amongst bacteria? See guys, when a bacterium multiplies, how does it multiply? By binary fission. So when a bacterium multiplies, obviously the genes are going to be passed on to the offspring, right? This is known as vertical transmission. But there is something known as horizontal transmission of genetic material also, all right, which we are going to cover under this broad heading of bacterial genetics, and we are going to understand three important phenomena wherein the exchange of genes is happening not from a parent bacterium to a daughter bacterium during division, but from one bacterium to another bacterium. Okay, not necessarily its daughter cell. So we are going to study transformation, the three methods of horizontal gene exchange. This itself is a question. Horizontal gene exchange, these are the three names: transformation, conjugation, and transduction. Okay, and transduction, right? So guys, before I start the topic per se, you need to understand some important phenomena. For instance, the very first one is homologous recombination amongst genes. So we have got, let's say, this yellow colored bacterium. Okay, this yellow colored structure is a bacterium, right? And as we are seeing, this bacterium has got this blue colored chromosome. Remember, bacteria don't have any nucleus, right? They have got a single circular double stranded molecule of DNA which serves as their genetic material. All right, so on this chromosome, as represented by the blue circle in this figure, we have got these genes A plus, B plus, C plus and D plus, right? Plus this bacterium also has got something else, the gene for Rec-A, which stands for recombinase enzyme. Okay, recombinase enzyme. All right, let's say this bacterium in its vicinity or neighborhood, it had some genetic material, right, known as exogenote, which now has entered this bacterial cell. Okay, and this exogenote has got these genes A minus and B minus. All right, and let's say A minus and A plus, the letters are the same, just with a difference of plus and minus to emphasize on the fact that these are homologous genes. Okay, so A plus, A minus and B plus, B minus. Okay, so what will happen, because these genes are homologous, there is a possibility that the genes can get exchanged. Okay, this kind of genetic exchange amongst genetic material is possible if two conditions are met with. Number one, the genes have to be homologous. For instance, over here we are seeing that A plus A minus pair and B plus B minus pair, they are homologous pairs, right? The second condition to be met with is the presence of the recombinase enzyme, guys, okay, which has been shown like this green circle. Now imagine, okay, when we are driving, okay, when we are on the road, imagine the traffic signal lights, the green signal signals you to move ahead. So similarly over here, the green color of the Rec A gene product is telling the bacterium actually that, okay, go ahead, you have my blessings, you have got homologous chromosome pairs, go ahead and get yourself exchanged, let's spice us let's spice up your life. So what happens now, this bacterium, because it is as I said, it's a blessed bacterium, blessed because of the presence of recombinase A. So next thing which happens is there is exchange of genes. As you can see, A plus and B minus, so A plus and B plus have come out and they have been replaced by A minus and B minus. Okay, there is no addition of genes, there is exchange of genes, and this kind of gene exchange, this kind of stabilization of genome is known as homologous recombination. Okay, now suppose this bacterium did not have a recombinase enzyme. Okay, then what would have happened? Do you think the recombination would have taken place even though the A plus, the A minus and B minus genes are homologous to A plus B plus? No, it would not have taken place. In that case, the exogenode which has entered this bacterial cell, it would have disintegrated. Okay, so as I said, both the conditions have to be met with, and this kind of incorporation of this exogenode into this bacterial, into this bacterial chromosome in the presence of Rec A enzyme, it is a method of stabilizing this genetic material. Okay, now these genes A minus, B minus could be very significant. They could be genes carrying they could be genes carrying information regarding some virulence factor, for instance some toxin, or gene for capsule production, or genes for resistance to certain group of drugs. Alright, so some change will happen in the life of this bacterium. Alright, this is known as homologous recombination. Okay, so guys, next is site specific recombination. Now let us see what happens in this case. Let's say this is a bacterium. Okay, now this is a bacterium with the blue colored chromosome with following genes A plus, B plus, C plus and D plus, right? And this bacterial chromosome has got these integration sites as shown over here in sky blue color. Alright, now imagine these integration sites are like a door, and it is at these integration sites that the chromosome is going to open up and it is going to incorporate some other genetic material. Okay, now that some other genetic material is this one, this circular molecule. This is another set of genes, and that also has got a door like structure shown by this purple color. So basically this set of genes shown in red color, what is this? This is some genome which has come from outside, and this purple door kind of a thing is the place at which this genetic material is going to open up, right? So what happens when both these doors open up? There is going to be addition or incorporation of the red genome into the blue genome. All right, so imagine the purple door has opened up. If I have to draw this, when it's something like this, suppose it has opened up like this, and the bacterial chromosome that also opens up like this. Okay, something like this. So obviously what happens? This end joins to this end and this end joins to this end as shown in this figure. Alright, so we are going to get this as the final product as shown over here. Now over here what happens? Extra genes have been added. Unlike homologous recombination where genes were getting exchanged, here genes have been added and not exchanged, and these genes need not be homologous, right? Unlike homologous recombination, right? So this is known as site specific recombination. This is known as site specific recombination. Why site specific? Because both the bacterial chromosome and this genome, the red genome which has entered this bacterial cell, they are going to open up at specific sites, right? And after the sites are opened, the joining of the chromosome is going to happen at these sites only. Okay, so integration sites these are. So this is known as site specific recombination. Okay, so remember there are two methods. The methods of stabilizing the genetic material are as follows. homologous recombination, and site specific recombination. In homologous recombination, the genes are homologous and recombinase A enzyme is needed. Okay, there occurs gene exchange occurs and not gene addition. In site specific recombination, the genes need not be homologous, right? Rec A enzyme is not needed, right? Over here new genes are added. In homologous recombination also, new genes are entering the cell, but we use the term exchange to emphasize on the fact that the total the total number of genes is going to be the same, unlike in site specific recombination where genes are getting added, so the number of genes is going to increase. All right, now guys, now that we have seen what are the methods of stabilizing the genetic material, that is homologous recombination and site specific recombination, let us understand the very first method of horizontal gene exchange amongst bacteria, that is transformation. Okay, now let's say we are going to discuss this in a series of pictures from left to right. All right, so we have got figure 1, where we have, in figure 1 we have this free DNA from some dead cell which is lying outside, okay, outside this yellow colored bacterial cell. Now this yellow colored bacterial cell has bacterial chromosome like this, double stranded. For the sake of simplicity, 2 circles have been drawn, right, to represent the bacterial chromosome in blue color. All right, the double stranded nature has not been shown. Okay, just remember there are 2 strands, right, of DNA. So right now we are going to understand this yellow bacterial cell as being a non-competent and rough, okay, and rough, that is non-encapsulated strep pneumoniae. Okay, so basically the bacterium in question is strep pneumoniae or pneumococcus, and let's say we know that pneumococcus is capsulated, but let's say this is a strain of pneumococcus which does not have the capsule. All right, now this pneumococcus in its neighborhood or vicinity, this free DNA is present which has come from some dead cell. Okay, so non-competent means that this bacterial chromosome right now is not permitting this free DNA to attach to its surface. Okay, let's say this free DNA wants to come inside this bacterial cell, but right now that is not possible because for this free DNA to enter the bacterial cell, the free DNA has to first attach to the cell surface. Okay, that is not yet possible in this cell. That is why this bacterial cell is said to be non-competent. But under certain growth conditions, what happens? This bacterial cell becomes competent. Why competent? It is going to express certain proteins on its surface to which the red genetic material is able to bind. Okay, so now this pneumococcus is said to be a competent cell which is able to bind the DNA, right? Next, what happens? Next, there is internalization of this red DNA, and then there is stabilization of certain genes by homologous recombination, which we just saw, right? And I want you all to notice carefully an important morphological change in figure 4. What you can see over here? You can see this nice capsule is now present in this bacterium, which means what? Which leads us to conclude that this gene which entered, the red gene which entered this pneumococcus, was the gene which was responsible for encoding the capsule. So now we are going to call this strain of Streptococcus pneumoniae as the smooth strain or encapsulated strain. We know that capsule is such an important virulence factor of pneumococcus. So this is how it has acquired the capsule. Okay. So in the first 3 figures from left to right, in 1, 2, 3, there was no faint yellow colored outline, right? Whereas in figure 4 we have got this faint yellow outline which represents the capsule. Now let us see the same thing in a little more detail. What actually is happening during transformation is, look at this. the double stranded DNA binds to the plasma membrane of the bacterium, right. This double stranded DNA is nothing but that DNA coming from the dead cell which is there in the in the neighborhood of this bacterium. Now next what happens, the DNA is cut by certain enzymes, right, and then some other enzymes break down the DNA strand. Okay, so one strand is only going to be left behind, and that one a protein is going to bind the DNA. The one strand which is going to be left behind over here, next it is going to bind to a protein and it is going to enter the cell. Okay, so the DNA replaces a portion of the bacterial DNA as occurs in homologous recombination, right, as we saw in the previous diagram, right. So this is known as transformation. Why it is called as transformation? Because obviously a transformation or change in the life of this bacterium has occurred, which previously was non-pathogenic or avirulent, but now has become pathogenic or virulent by virtue of possession of the capsule, right. Now guys, this phenomenon was of transformation was discovered by Griffith. It was discovered by Griffith in Streptococcus pneumoniae or pneumococcus, okay, and it is also observed in Haemophilus influenzae. All right. Now look at this picture, transformation. Now imagine over here we have got the rough strain, that is the non-virulent strain. Why non-virulent strain is called as rough strain? The strain is non-virulent because it doesn't have capsule. Because it doesn't have capsule, the colonies are going to be rough, right. On the other hand, in a virulent strain which is going to have the capsule is going to be responsible for the smooth colonies, right. So the rough strain over here, which is harmless, is being shown by the color of peace, that is blue color. So imagine when we inject the rough strain into the mice lives, okay, the mouse lives. But when we inject the smooth strain or the virulent strain, which is dangerous, so red in color, and as you can see a capsule also has been drawn over here, when this dangerous virulent strain is injected into the mice, the mice is expected to die. But when we kill the smooth strain by heating it, so that is known as heat killed smooth strain, then also the mouse is expected to live because we have killed the strain, we have killed the bacterium, so it will not cause any disease. But surprise observation is there when we inject the mouse with a mixture of the rough strain, the live rough strain, and the heat killed smooth strain. Okay, so this is the rough strain which is live, and this is the smooth strain which has been heat killed. What do we expect? We know that rough strain is avirulent, it will not kill, and smooth strain is also the smooth strain itself is killed, so it anyways cannot kill. But when we expect the mouse not to suffer at all, we expect the mouse to live. But to our surprise, what happens when we inject this mixture, the mouse dies. Okay, the mouse does not live, it dies. It means that see guys, the smooth or the virulent strain was anyways we killed it, right, so it could not cause any death. But when we mixed it with rough strain which was alive, that alive strain was able to kill this mouse. It means that some change in the rough bacterium, the rough live avirulent bacterium must have happened when we mixed it with the smooth strain. All right, and that is nothing but this whole diagram which we have discussed already. Okay, so imagine this yellow bacterium, this is the rough live strain, and this free DNA shown in the maroon or red color, this is coming from the dead, that is the heat killed smooth strain. So that is what was observed by Griffith. This experiment in mouse was done by Griffith. So guys, this is the very first method of genetic exchange in bacteria, horizontal genetic gene exchange, transformation. All right, now guys, next we are going to discuss conjugation, for which you need to know a specific type of plasmid known as F factor or fertility factor, or F factor, okay, or fertility plasmid. Already I have spoken about plasmid in one of the video modules on general microbiology, right. Remember, P for plasmid, P for prokaryotes. Plasmid is present in prokaryotes, that is bacteria, not in all the bacteria. because it is not present in all bacteria. It is not an absolutely essential thing for bacteria to survive, but whatever bacteria have plasmid, they usually have some or the other advantage over the other bacteria which don't have plasmid. What is plasmid actually? Plasmid is nothing but, plasmid is nothing but double stranded DNA only, just like the bacterial chromosome, but it is smaller. Okay, and plasmids are what? They are autonomous. They are autonomous self-replicating entities. Their replication is independent of replication of the bacterial chromosome. So over here we are going to discuss one type of plasmid known as fertility factor or F factor, which takes part in conjugation. Okay, so it looks like this. We have got a site over here in this F plasmid known as ORIT, that is origin of transfer. Now what happens over here? A single strand breaks over here. Plasmid is also double stranded. At the ORIT site, that is origin of transfer, one of the two strands of the plasmid will break, and this is where the transfer of the single strand begins, as we are going to see in the next diagram. Okay, the other strand which was not cut, it remains and it is quickly restored to double strandedness. Right. So what happens, we are going to see in a while that when one plasmid from one bacterial cell is supposed to enter the other bacterial cell, for this to happen, one of the two strands of the plasmid is cut, and that cut strand is going to go to the other bacterium through a tunnel called as conjugation tube. Okay, that is what this means. And so the donor bacterium which is donating this plasmid is going to be left with just one strand of the plasmid, but the donor bacterium is going to replicate the complementary strand. Okay, so the donor has got nothing to lose, and the recipient which is receiving one strand, that also the recipient cell will also replicate. The recipient cell also will synthesize the complementary strand, right? So the donor remains donor only, whereas the recipient becomes donor, meaning the recipient cell in future can again pass on this plasmid to some other cell. Okay, so a plasmid must have this region to be transferable by conjugation. Okay, so just remember, ORIT is the region where a cut is made in one of the strands of the plasmid, right? And then we have got just like we saw in site specific recombination, there are insertion sequences. Let's say if this plasmid wants to get integrated into the bacterial chromosome, then it should open up, and that opening up side is nothing but the insertion sequence, right? And then because I said that these are self-replicating entities, suppose this plasmid wants to multiply, right? So for that also a site is needed, an area is needed known as ORIV, that is origin of vegetative replication, right? So as I said, the insertion sequence, this is the place, this is the site where site-specific recombination may integrate the fertility factor into the bacterial chromosome, and when this happens, we are going to call it as an HFR, that is high frequency of recombination. I'll come to that in detail in a few minutes, right? And then this fertility factor also has got different genes. Remember, plasmid is nothing but DNA only, na? It is double stranded DNA, it is going to have genes, okay? And that one combination or amalgamation or group of genes is tra-opron, and it has got different genes. The most important ones are the genes which encode for sex pili, for the metabolism of this conjugal DNA, etc. Okay, so this is how a fertility factor plasmid or NF plasmid looks like. Now let us see the mating types in bacteria, guys. Now imagine we have got this bacterium which is not having any plasmid, no F factor or F plasmid, so it is going to be referred to as F minus. Alright, so this is going to be labeled as the recipient bacterium. Okay, recipient bacterium. Okay, now there is another bacterium which is referred to as donor. Why? Because it has got something to donate. What it has? It has got the F plasmid. Okay, this is the F plasmid as indicated by TRA. Remember, TRA is the name of the opron or the group of genes which we said is present in the F factor. Okay, so this is known as the F plus cell. Okay, it is called as donor because it has something to donate. What it can donate? It can donate the F plasmid, right? And it can also happen that in some bacterial cells the plasmid can get inserted into the bacterial chromosome, and in which case we are going to call it as HFR. This is known as high frequency recombinant. This bacterial cell is also said to be a donor cell because it still has the plasmid. Doesn't matter that the plasmid is now inserted into the bacterial chromosome, it still can donate at least part of the some genes from this plasmid into some other cell, as we are going to see in a few minutes, right. So these are the three kinds of bacteria: F minus, F plus and HFR. Now let us see how how these bacteria conspire amongst themselves to bring about gene exchange. So let us see what happens when an F plus and F minus bacterium, they undergo crossing or mating with each other. Okay, so now let us see. So this is we have got an F plus bacterium. I would emphasize highly that you draw this diagram. These are schematic diagrams only. All right, keep drawing this as you are watching this video. So this is the F plus cell, guys. Okay, the bacterial chromosome has got these genes A plus, B plus, C plus and D plus, right, and you have this plasmid, and in the vicinity, in the neighborhood, you have got the F minus cell, which is poor because it doesn't have any plasmid. Now this F plus cell is able to produce, or it is able to express a tube like projection. This is nothing but made up of proteins. This projection is known as sex pilus. You know why? Because of the presence of this plasmid. Now let us go back and revise what we learnt in the fertility factor. Remember, the traopron in the fertility factor had got genes to form the sex pilus also. Okay, so guys, the F plus cell, because it has got the plasmid, it can form this sex pilus, and this sex pilus is then going to form this tube or bridge. This is known as conjugation tube or bridge, which is going to connect these two bacteria now, right. So what happens? Imagine, now let me just explain this schematically. Imagine you are eating Maggie, and you have got the Maggie. Let me use yellow color for Maggie. The Maggie, there are two strands of Maggie on your plate like this, intertwined with each other. Now let these two strands of Maggie analogous to the plasmid. Okay, so we have got Maggie or the plasmid, and this is your plate. Now your friend is really hungry, and you decide to now, even though you have just two strands of Maggie to eat, but still, because you are a very good friend, you are determined and motivated to help your friend who is also hungry. Okay, so what do? You just donate one of your Maggie noodle strands to your friend. Okay, like this. Now let me erase one strand from your plate. Okay, so let us say this is your plate, this is your friend's plate. Both of you are left with what? Both of you are left with now one Maggie strand each. But God is kind to both of you, because you are still able to manage to cook another strand of Maggie. Alright, maybe some other friend helps you. So what happened? Your intention was good, you wanted to help your hungry friend. So God came to your rescue also. Alright, so you are not going to remain hungry. You will still be having two Maggie strands even after you donated one Maggie strand to your friend. And your friend is very, very lucky because your friend is being helped by you and by somebody else also who helped in the creation or manufacture of the second Maggie strand. Okay, so you didn't lose anything, and your friend also gained a good two Maggie noodle strands. Okay, so similarly over here also, when this conjugation tube forms, what happens? One of the two strands in the plasmid is going to undergo cutting, as we just saw in the structure of the F plasmid, okay, at ORIT region. So when the conjugation tube forms, some change will occur in the plasmid, and what is that change? That change is nothing but cutting of one of the strands of the plasmid, so that one strand goes to this cell, the F minus cell. So what will happen? This cell, the donor cell, is going to be left behind with one strand of the plasmid, and the recipient is going to gain one strand. But both the donor and the recipient are going to synthesize the complementary strands of their single strands, right? So what happens? The F plus cell has got nothing to lose. The F plus cell will remain F plus only, it will remain an F plus, and the F minus cell now becomes F plus because it has acquired a plasmid, right. So this is F plus cell with a new plasmid, but no new bacterial genes, meaning no change in the chromosome, in its chromosomal genes has happened. It just has, now it has come to acquire the plasmid, that's all. Okay. So at the end of F plus by F minus conjugal cross, what happens? The F plus remains F plus only and F minus becomes F plus. Okay. Then guys, next we have HFR by F minus conjugal crossing. Okay. How did we define HFR? That is high frequency of recombination. It is a cell where the F plasmid integrates. It integrates with the bacterial chromosome. It integrates with the bacterial chromosome. Alright. So this is HFR cell. As you can see, the red colored plasmid is integrated into the bacterial chromosome, and this is F minus cell, F minus. Okay. So what happens? Because the HFR has got the plasmid, doesn't matter that the plasmid is not separate, it doesn't matter that it is attached to the bacterial chromosome. By virtue of presence of plasmid genes, a conjugation tube, this is the conjugation bridge or tube, is going to be formed between both these cells. And what happens now? Look at this. Now I am magnifying or zooming in this diagram. Please listen to me very, very carefully. Now guys, this is the ORIT or the origin of transfer. Alright, so over here, once the connection is formed between both these cells with the help of the conjugation tube or bridge, then a cut or nick is going to be created in one of the strands of the plasmid at ORIT site. And I want you all to notice what is behind ORIT. Behind ORIT, you have got gene number 1 followed by 2, 3, 4, which are not marked in this diagram. Let's say until 7, the plasmid has got genes from 1 to 7. Okay. So the gene which is nearest to ORIT is gene 1. Imagine ORIT is the engine of a train, and the very first dabba of the train is the compartment number 1, and behind compartment 1 is present 2, 3, 4. So compartment 7 is way behind the engine, that is ORIT. Okay. So after the conjugation bridge has been formed, what happens? The ORIT is going to be, the cut was made at ORIT. So from this point onwards, the broken strand of the plasmid is going to pass through the bridge and enter the recipient cell. Okay, this one. So ORIT is over here. This is the ORIT region of the plasmid gene, and behind it is present gene number 1, right. So gene number 1 to 7 come to, they land up in this daughter in this recipient bacterial cell. And who was behind 7? Who was behind gene 7? Behind gene 7 were the bacterial chromosome genes in order A plus, B plus, C plus and D plus. Okay. Now because A plus gene is nearest to gene 7 of the plasmid, there is a high frequency of transfer of A plus gene, and then lesser frequency of transferring B plus gene, and even lesser frequency of transferring C plus gene, which is further at a greater distance, like that. Okay. So theoretically it is possible that the entire bacterial chromosome can be transferred like this, but that doesn't happen because this bridge has got a short half-life, it breaks down soon. So that is why HFR cell is called so, because there is high frequency of transfer of certain genes only, okay, whenever this mating process is happening. Okay. So what happens next? Let's see. Let's say next, in the next step, this bridge gets broken down. So what do we see? We observe that ORIT came into this this cell, the second cell, and behind ORIT was gene 1 to 7, not shown here. Then A plus, B plus, C plus, but D plus was not transferred. Okay. Now next what happens? See, even though a cut was made in the plasmid and some genes had now have gone to this cell, to the F minus cell, still the HFR cell is, because of because it has got the nucleotides, it is going to synthesize the complementary strand. Okay. So HFR will remain HFR only. So HFR is going to, it has got nothing to lose. Okay. It is like Tata money, it has got a lot of it has got nothing to lose. Okay. No money to lose, no genes to lose, right. Now let us focus at this F minus cell. Now what happens? Which genes came? See over here, ORIT A plus, B plus, C plus. All right. Now this F minus bacterium had got which genes with itself? A minus, B minus, C minus. So basically A plus, A minus, B minus, B plus, and C plus, C minus, all these are homologous genes. Okay. Now there can be there is a possibility that homologous exchange happens in this F minus bacterium if it is producing the recombinase A enzyme. All right. So we are going to end up with F minus cell with new genes. Let's say in this diagram A plus and B plus genes have been shown, but C plus gene could also be incorporated, right? So don't worry about it. Don't think why C plus was not exchanged. It also can get exchanged. It's just that in this diagram we have shown the exchange of only A plus and B plus. Okay, C plus also could have been exchanged. Okay, so don't worry about that. Okay, now no sex change occurs in F minus cell. This remains F minus only. This still remains F minus. You know why? Because it has got only certain genes of the plasmid. It has not come to acquire the full plasmid, the entire genes of the plasmid. Okay. So guys, that's about HFR by F minus conjugal cross. Remember, at the end of it, HFR remains unchanged, but F minus does acquire some change, but it still remains F minus only. Alright. So guys, this is about conjugation.